Okay, so this is part 2 of the cell. You'll be able to access this tomorrow, I hope, if all goes well, and I don't have to repeat this 5 or 6 times. All right. Last time I talked briefly about the cell, and just as a quick review, complexity, compartmentalization, just write compartments, the dynamic nature of the cell, and the fact that the cell is an energy transformer. We'll get into a little bit of that today. All right, so let's draw a picture of the nucleus here, and the nucleus is about 5 microns in diameter on average. It contains material known as chromatin. Chromatin, chromos meaning color, because the nucleus tends to take up dyes, and the chromosomes were actually analyzed with dyes, and the particular pattern of dyes on a chromosome can be used to identify them. As you probably know, you have 46 chromosomes, 23 from mom, 23 from dad. Each of those sets has about the same number of nucleotide base pairs, about 3 billion for each set. So together, the genome within the nucleus of a human being, about 6 billion base pairs. You're probably wondering, what in the world is he talking about? Well, those A, T, C's, and G's. Adenine, thymine, cytosine, and guanine. These are the base pairs that in a particular sequence create a coded message for genes. Now I'm going to represent the double helix here symbolically, okay, with that. Now, of course, it's not that that big. All right, if you could take the DNA from one cell of a human being, from each one of those 46 chromosomes, and lay them end to end, it would be about 2 meters or 6 feet in length approximately, but only about 2 nanometers in size, extraordinarily thin. So thin, in fact, that you couldn't even see it. If we made, however, that molecule large enough so that you could see it as a thread, perhaps 1 millimeter in diameter, then the DNA proportionally would be a couple of thousand kilometers in length. DNA is packaged in the nucleus in a very specific way, and by proteins known as histones. There are 4 basic types of histones that make up what's called a nucleosome. The DNA double helix is actually wrapped around this body, and that is the first order of the packaging of DNA. It's further then wrapped up again and again in a very complex way to form the bodies that are visible during the stages of mitosis that we call chromosomes. But during interphase and during most of the lifespan of a cell, the chromatin is unraveled, so the DNA is exposed to enzymes that are inside the nucleus. Now, within the nucleus, there's also a dense body known as the nucleolus. And most of the time there's one of these bodies, but sometimes there can be 2, and some groups of organisms are actually known by this characteristic, the diplomonads. Giardia, which we sometimes suffer around here when we drink water that hasn't been boiled out of a stream in the Nevadas, and you come back and you've got severe diarrhea. Well, you've been infected with one of these protozoans. In any case, the nucleolus we know now is basically ribosome production organelle, if you will. I don't like to call it an organelle, but it's the site of manufacture of ribosomal RNA. And this is going to come into the story here in a minute. All right. The nucleus is perforated with these tiny pores. Okay? And through these pores, then substances can go back and forth from the cytoplasm into the nucleus. And remember, the nucleus is actually a double-membraned organelle. So there's two bilayers that surround it. And the outer bilayer is actually continuous with the endoplasmic reticulum. So I'm going to attempt to draw that here. Okay. The endoplasmic reticulum is a very convoluted, flattened, membranous system, which looks like something like this in electron micrographs. Okay, and the spaces within these flattened stacks of membrane are called cisternae. Cisternae, that's plural. Okay, so you know, I'm going to point to a cisternal space there. Okay, and on the surface, I'm going in red now to put ribosomes. These are sometimes described as organelles, but they're not surrounded by any membranes. And it's best to think of these things as molecular machines. Life invented nanotechnology more than 3 billion years ago when it came into existence by some process we don't completely understand as yet. But the nanobots are here, and they've always been here. They're in every one of your cells and in the cells of every living thing on this planet. Ribosomes come in two flavors, basically. Those found in eukaryotic cells, which are a little larger than the ones found in prokaryotic cells. Now remember, prokaryotes are the bacteria and the archaea and the cyanobacteria. Those organisms do not have a nucleus. All eukaryotes do have a nucleus. Okay, and their ribosomes are slightly different. But all ribosomes have two subunits, a large subunit and a small subunit. Thank God, that part is easy. And the ribosomal RNA, the rRNA, which is made in the nucleolus, combines with proteins that are produced out here to form the ribosomes themselves. So when I described this earlier as a ribosomal factory, that's pretty much correct. So proteins that will assist in the construction, so to speak, of ribosomes move into the nucleus. The ribosomal RNA combines with those proteins or polypeptides, and the small subunit and the large subunit are actually formed within the nucleus. Those subunits move out of the nucleus and then combine when they meet up with messenger RNA. Okay, so that's another form of RNA. I'm going to write it here. Messenger RNA. All right, there's actually 3 forms of RNA. Might as well tell you the third form is called transfer RNA. Okay, so a particular enzyme produces now a copy. And there's a lot of detail here, and I'm just giving you the broad picture. Okay, but a DNA-dependent RNA polymerase. RNA polymerase. It's going to polymerize. All right? And so I'm going to draw that here, and I'll make the transcripts, or the messengers, blue in color. So that large attaches to DNA and produces a copy of a section of DNA that is the message. Okay? Now, messenger RNA, ribonucleic acid, is slightly different from DNA, deoxyribonucleic acid. So I'm going to outline those basic differences now. As you probably already learned, DNA contains these 4 bases, adenine, thymine, cytosine, and guanine. RNA is slightly different. It has adenine, but no thymine. Instead, it has uracil, it has cytosine, and guanine. And unlike DNA, which is a double helix and a more stable molecule, RNA only has a single. It's not a double helix. So that's why I've drawn it here as just a blue line. It diffuses out of the pores of the nucleus, and then when it collides essentially with a ribosome, it forms a complex. So here I've shown, I don't know if you can see it, it's very small, sorry, the small subunit and the large subunit. The messenger RNA finds a cleft in there, and these ribosomes are essentially tiny factories that are going to convert the message taken from the gene by messenger RNA into the proper sequence of amino acids to produce the particular polypeptide or protein coded by that particular gene. And so as the ribosome moves along the messenger RNA, it reads that code and then produces the specific protein. Here I've shown the protein, and as it goes down, the protein will grow. Other ribosomes will attach, and as it goes down, the protein will lengthen. Okay? Remember, proteins are composed of amino acids. There's about 20 of them that are necessary for life and found in all living things. All right, now if this messenger attaches to a ribosome here, the protein formed will thread itself through tiny pores into the endoplasmic reticulum. And protein that forms in this way is destined for secretion. And I will describe that process and how those molecules destined for secretion will interact with the Golgi apparatus in the next video. I've got about a minute left. Let me also say the endoplasmic reticulum produces produces its own structure. So the proteins necessary for building itself, along with the phospholipids and glycolipids necessary for its membranous structure, will be produced there. By the way, there's 2 types of endoplasmic reticulum, those with ribosomes. That's called rough endoplasmic reticulum, and then those without. And the endoplasmic reticulum without is just called smooth ER. And it actually looks a little different too in electron micrographs, pictures taken with an electron microscope. Instead of being flattened membranous, very complex spaces, the smooth endoplasmic reticulum is more like tubules, although all endoplasmic reticulum is continuous with each other. All right, so I'm going to cut it there. I hope that was illuminating or illuminating.